Okay, imagine this, right? Imagine having the power to just rewrite the very code of life, like actually going in and fixing genetic diseases, creating super crops, maybe even like sculpting traits in future generations.
Hmm. So it's like something out of a movie, isn't it?
Right. It sounds like pure science fiction, but what if I told you it's not? This is actually happening like right now with this technology you've probably heard about. It's called CRISPR.
Yeah, CRISPR is definitely having a moment, right? It feels like it's everywhere.
It is. And today we're going to take a deep dive into it. Welcome to Dansplain, by the way. I'm Edward.
And I'm Lily. So, yeah, today it's all about CRISPR.
Sure.
Clustered, regularly interspaced, short palindromic repeats.
Catchy,
Real mouthful. But really, CRISPR is more than just, this cool new tech. It's transforming biology, but it's also sparking a global conversation about, well, about a lot of things. Ethics, the future of medicine, all kinds of stuff.
Yeah, it's big. And you, our amazing listeners, you sent us some seriously interesting stuff on CRISPR. And our mission today is to unpack all of that, to really get to the heart of why this technology is such a big deal. We want to have those like aha moments where it all clicks into place.
Totally. CRISPR is such a fast-moving field. There's always something new to learn. So where do we even begin? I mean, it's interesting. The discovery of CRISPR wasn't like this eureka moment in some fancy lab, was it? The origin story is a bit more unexpected.
Yeah, it really is. So picture this. It's 1987. A group of researchers in Japan, they're studying E. coli bacteria, and they find these weird repeating sequences in the DNA.
It was kind of a head-scratcher at the time, wasn't it? Just this strange repeating pattern. No one knew what it meant.
Exactly. For years, those repeats were just a mystery. Then, fast forward to the early 2000s, a Spanish scientist named Francisco Mojica, he's looking at these repeats and he starts to see the bigger picture. He realizes these same patterns are popping up in all kinds of bacteria and archaea.
He really was the first to see the potential there, wasn't he? In 2000, he even gave these sequences their now famous name, CRISPR. And here's the really amazing part. He suggested these CRISPR sequences might actually be part of a defense system for bacteria, like this hidden immune system, just waiting to be discovered.
That's incredible. Sometimes the biggest discoveries come from the most unexpected places, don't they? But how did they figure out exactly how this bacterial defense system worked? It seems pretty complicated.
Yeah, it's incredibly elegant, really. Over the next few years, Mojica and a bunch of other scientists, they dug deeper and deeper into CRISPR. They proved that CRISPR, along with these special proteins called Cas proteins, it all works together as an adaptive immune system in bacteria. Think of it like a microbial memory bank. When a virus attacks the bacterium can actually take a little snippet of that virus's DNA and store it in its own CRISPR sequence. Then, if that same virus attacks again, bam, the bacteria recognizes it and uses those Cas proteins to chop up the virus's DNA, basically destroying the invader.
So bacteria have their own little ninja warriors, huh? Pretty sophisticated for such tiny organism.
Right. Nature's full of surprises. But here's the thing. How did we go from like, oh, that's cool, bacteria have a cool defense system, to actually using that system to edit genes in, like completely different organisms, even ourselves? That seems like a huge leap.
Yeah, definitely a big leap. So this is where it gets really interesting. In 2012, two scientists, Jennifer Doudna and Emmanuelle Charpentier, they did something groundbreaking. They showed that you could reprogram the CRISPR-Cas system. They figured out how to design like a custom guide, a piece of RNA that would tell the Cas9 protein, okay, go cut the DNA right here at this exact spot.
That was the turning point, wasn't it?
Yeah.
That's when CRISPR went from being a cool observation in bacteria to a powerful tool that we could control. It's no surprise they won a Nobel Prize in chemistry for that work in 2020. That was huge.
So now we have these molecular scissors, super precise, and we can program them to cut wherever we want in the DNA. That's amazing. But I bet getting that to work in, like, the cells of plants and animals, that wasn't easy.
Right, it's a lot more complicated than just working with bacteria in a test tube. But scientists saw the potential, and they moved fast. Just a year later, in 2013, two other scientists, Feng Zhang and George Church, they both independently figured out how to make CRISPR-Cas9 work in human and mouse cells.
That's amazing. So suddenly you can edit genes in any living thing, potentially. I mean, what doors did that open up?
So many doors. The medical applications alone are mind-blowing, especially for treating genetic diseases. And it didn't stop there. By 2015, people were already using CRISPR to edit plants, creating mushrooms that don't brown, for example. Even animals were being genetically modified with CRISPR, all for research purposes.
Wow. So CRISPR is not just for humans then, it's for all kinds of organisms. It's really changing everything. Okay, but let's break this down a bit more for our listeners who might not be like molecular biology experts. How does this CRISPR-Cas9 system actually work? How does it find the right spot to cut in like this massive amount of DNA?
It's a good question. Imagine this. The Cas9 protein, it's like a pair of really fancy scissors, but it also has a built-in GPS. And the GPS in this case is a piece of RNA, the guide RNA we talked about. This guide RNA is designed to match the exact DNA sequence you want to edit. It searches through the DNA, and when it finds a perfect match, it locks on. And then the Cas9 protein attached to this guide RNA, it makes a cut in the DNA right at that spot.
So the guide RNA is like the address, and
Yeah.
Cas9's the delivery guy, right?
Yeah.
Super precise, super efficient. But I think I read somewhere that Cas9 isn't the only protein involved in CRISPR, right? There are other Cas proteins too.
Absolutely. Cas9 is kind of the celebrity of the CRISPR world, the one everyone knows. But it's just one of many Cas proteins that have been discovered. These different Cas proteins like Cas12 and Cas13, they all have their own unique ways of working. For example, Cas13, it's really interesting because it targets RNA instead of DNA.
So instead of cutting the DNA, it cuts the RNA. What does that even mean?
It opens up a whole new set of possibilities. Instead of making permanent changes to the DNA, you can use Cas13 to, say, temporarily turn off a gene by targeting its messenger RNA.
Oh, okay. So it's like you're not rewriting the whole book. You're just like putting a sticky note on a page to skip over it for a bit. That's really cool.
Exactly, and it doesn't stop there. Scientists are constantly developing new CRISPR techniques. There's base editing, for example, which lets you change just a single letter in the DNA code without actually cutting the whole strand. This is like super precise, and it could help avoid some of those unintended consequences that people worry about with CRISPR.
So it's like a molecular spell checker, just finding and fixing the typo, not rewriting the whole sentence. Very cool.
And then there's prime editing, which is even more advanced. It's like a molecular word processor. You can insert, delete, or even replace entire sections of DNA with incredible accuracy. So the CRISPR toolbox, it's not just powerful, it's constantly evolving and getting better.
This is amazing. I mean, it sounds like CRISPR can do practically anything. So where are we seeing it being used right now? What are the real-world applications of CRISPR?
Oh, the applications are popping up everywhere. In medicine, like we mentioned, clinical trials are underway for inherited genetic disorders like sickle cell disease and beta thalassemia.
Wow, so potentially life-changing for people with those diseases.
Right. The goal is to actually fix the faulty genes that cause these conditions, which
Yeah.
could essentially cure them. And CRISPR is also making waves in cancer research. Scientists are using it to engineer immune cells, those CAR T cells, to make them better at hunting down and destroying cancer cells.
That's huge. So we talked about medicine. What about other areas like agriculture? I know CRISPR has some pretty amazing potential there too.
Oh, absolutely. In agriculture, CRISPR is being used to develop crops that are more nutritious, resistant to pests, and able to grow in harsher conditions. Remember those non-browning mushrooms we talked about? That's just one example. There's also drought-tolerant rice, disease-resistant wheat. The possibilities are endless, really.
So potentially CRISPR could help us feed a growing population even in a changing climate.
Exactly. And it's not just food production. CRISPR is revolutionizing biotechnology as well. Scientists are using it to engineer microbes for a whole range of applications, like producing biofuels, creating new drugs, even making sustainable materials.
So it's not just about treating diseases or improving crops. It's really transforming how we make things and how we interact with the natural world. Amazing. But I know no technology is perfect. What are some of the challenges, the limitations that CRISPR still faces?
You're right. Even with all its potential, CRISPR isn't a magic wand. One of the biggest concerns is off-target effects. That's when the Cas protein accidentally cuts the DNA in the wrong place, somewhere it wasn't supposed to. This could lead to unintended mutations, which could be harmful.
That makes sense. You want to make sure you're only changing what you intend to change.
Exactly. Another challenge is delivery. Getting the CRISPR system into the right cells in the body in a safe and effective way, that's still a hurdle. Scientists are working on different delivery methods like using nanoparticles or modified viruses as carriers.
It's like a targeted missile, right? You need the right delivery system to hit the right target.
Right. And then there's something called mosaicism, which can be a problem when editing embryos. This basically means that not all cells in the embryo get edited, so you end up with a mix of edited and unedited cells. That's a problem because it makes the outcomes unpredictable.
Okay, those are some pretty big challenges. So, looking forward, what's next for CRISPR? What are some of the exciting possibilities on the horizon?
Well, there's so much happening right now, it's hard to keep up. Scientists are constantly refining the technology, making it more precise and efficient. They're developing new Ks proteins that can target different parts of the genome. There's a lot of research on improving delivery methods, making sure the CRISPR system gets to the right place.
So basically, CRISPR is just going to keep getting better and better.
Absolutely. And the potential applications are just mind-boggling. We might see cures for a whole range of genetic diseases, from cystic fibrosis to muscular dystrophy to Huntington's disease. CRISPR could revolutionize cancer treatment, making immunotherapy even more effective. And it could help us combat infectious diseases in new ways.
So we're talking about a future where we can potentially cure some of the most devastating diseases, feed billions of people, and even reshape the natural world around us. That's a lot of power.
It is a lot of power. And with that power comes responsibility, right?
Yeah.
We need to think carefully about the ethical implications of CRISPR, about how we use this technology responsibly and make sure it benefits everyone.
That's a crucial point. We've covered a lot today, from the origins of CRISPR in bacteria to the incredible potential it holds for the future. It's been a fascinating journey, and we hope you, our listener, have learned a thing or two about this revolutionary technology.
Totally. It's a story that's still being written, and we can't wait to see what happens next. This is definitely just the beginning.
Absolutely. And if you found this deep dive into CRISPR as fascinating as we did, and you want to explore more of the cutting-edge science and technology that's shaping our world, well, you know what to do. Subscribe to Dansplain wherever you get your podcasts.
And for the latest episodes, in-depth articles, our curated list of partner products, and much more, visit us at www.dansplain.xyz.
Until next time, keep those brains buzzing.